Hello, my name is Julian. I'd like to thank the organizers for scheduling and taking care of the logistics of the phenomenal event that the workshop is. And I'd like to use this opportunity to advocate a bit about for the somewhat unreasonable efficiency and potency and phenomenal promise of methods inspired by statistical mechanics, namely energy landscape analysis and phase diagram analysis, especially when combined with appropriately adapted methods for obtaining shared alignments between different, systematically differing in neural dynamics subjects or even cohorts. The first of the two methods, namely energy landscape analysis, essentially draws on what's called an Ising model, which originally comes from statistical physics, to be even more specific from statistical ferromagnetism. And it was developed as far back as, I think, in the 1920s or 20s by Ernst Ising, whose picture from 1925 you can see here. And one of the reasons, arguments, I'd like to present for in favor of the methods is how universal and conveniently adaptable they are to analyzing about any dynamical system whose time series, whose dynamics we can meaningfully discretize, more specifically binarize. And this is very well illustrated by the broad, in terms of areas of application selection of the domains of applications which use, if not exactly Ising model, then very much adjacent methods, including domains of social science, which I think speaks to the diversity of the methodology. In our study, we developed and implemented several methods, including ones drawing on the Ising model, by using functional ultrasound data of mouse models of autism, namely rodents which were modified using Krylov recombinant genetic modification so as to best reflect the autism symptoms that appear in humans. And in terms of more technical aspects of the data, we operated on 54 brain regions, which were hemisphere coded. So we had 27 left and 27 right alternatives of the same region that was recorded from the whole brain. And the remaining aspects of the data include the temporal resolution or sampling rate of recordings of about 1.67 hertz, 0.6 seconds per frame. The most vanilla / general formulation of the Ising model that we use, and that's most commonly used, is the pairwise maximum entropy model, which essentially has two aspects to it. So the first one being, say, similar or analogous expression responsible for defining how probable any given dynamical or activity configuration pattern is in the brain. Namely, that's either the P or E, corresponding respectively to the probability of the state or to its energy. And the second most important aspect is the partition function, which could be familiar to people who come from a place of statistical physics. And this essentially forces us to normalize our P, so essentially calculate the probability of every possible region, every possible activation pattern, And this exactly is why the method, even though it's somewhat simple in terms of formulation and implementation, gets very expensive computationally. Actually, it's prohibitively computationally exploding, let's say, because if you were to calculate the exact formulation of the Ising model or the PMM method, we'd have to look at a state space of n to the power, of 2 to the power of n, where n is the number of our nodes in the dynamical system we operate on, or the number of brain regions we want to record from. And as you know, it explodes or grows very fast exponentially, for that matter, with any additional n. And because of that, we move to the second defining or one of the most important aspects of the work, which is applying, modifying, and stitching in an original way several methods for reducing the dimensionality / attaining a directly comparable shared alignment or representation, if you will, of the time series of the mice we had in the data. And there are, there are two motivations or key driving factors which determine that we want to, or actually need to use the dimensionality reduction method. First one being the pragmatic/standard one, namely that the method otherwise would be infeasible to use. So that's the exploding state space part. But the aspect which we cared more and which we actually developed more in our study was the thing of optimal representation of the dynamics of a mice. So the goal, put in broad strokes, was to obtain a shared representation of the time series of all the mice for each of their respective brain region, so that the reduced time series and reduced data frames of every one of the mice would be reduced efficiently. So we would look at a efficient or effective reduction in the number of dimensions of each such data. And the other aspect we cared about was besides direct comparability with mice, was the retaining of the key idiosyncratic or characterizing aspects of the time series of each mouse, which is somewhat self-contradicting if you think about it. And that's why we also developed, actually improved some of these methods so that we carefully balance how much we retain the particularities of any single subject, while also striving for a directly comparable universal latent representation of each of them on the population level. One of the details or technicalities which we developed and implemented for this purpose was a novel balanced fitness function, so a combination of many different metrics, sometimes shared by all the dimensionality reduction methods, sometimes specific to any one of them, and by weighting in a controlled manner and in general using different metrics which describe different aspects of the performance of any of these methods, we assure that none of the aspects, especially if it's exclusive to only one or not all of the methods, dominates. Just to briefly mention the main results or outputs of such workflow, on the left we can see the general, so shared for the entire population of all the mice, mapping of for the SRM model, so shared response model, from what we have on the left, the original brain regions, to what we have on the bottom of the matrix, the observed or resulting latent components. So depending on the phase sign of the time series, the values are either positive or negative, but essentially what you care about is the absolute value of how much any given brain region contributes to the shared representation for any given latent component. And on the right you can see one specific mouse whose time series were reduced using the common model that was obtained in a truly data-driven manner and which is represented on the left. As a quick side note, we've also tinkered with methods which were more quote-unquote interpretable. So the one on the left represents clustering of the time series which was purely driven by neuroanatomy per the Allen Institute Atlas, I think. And the one on the right, on the other hand, was driven by the similarity of the functional time series as measured by different correlation measures. But the issue is that both of these methods and at large methods which are function-driven and not some abstract projections into lower dimensional spaces do not yield sufficient reduction in the number of dimensions, which needs to be pretty low given the specificity of the methods we use. However, once we get all of that out of the picture, we are looking at a set of very attractive, in the sense of the results and outputs, metrics, deliverables, visualizations of the key energy landscape analysis process are very interpretable. That's one thing. And because and thanks to the previous instrument, let's say, that the data for each mouse was directly comparable across the cohort, we can also directly compare the resulting metrics or descriptors and all these results across the rodents, even though these results are produced on a level of any individual mouse. And we also can see here, sorry. It doesn't work. Anyway, top right, we can see the at-large process of obtaining the minima. So at first we feed the data to the models shown before, so the formulation of the pairwise maximum entry model, and then by using a disconnectivity graph, we identify the actual attractors of the energy landscape, so the configurations of activity patterns of the brain or about any dynamical system that occur the most frequently, and we also collapse very adjacent, very close neighboring states so that we don't end up with attractors that are just clustered all together. And a bit higher up in the rightmost visualization, we're looking at a mapping from the actual brain regions, or in this case, latent representations, features of the data, which we have on the left, to the identified attractor states, so the most frequently occurring configurations of brain activity. And why this in particular is so interesting and so useful is that by having all the results which we see on the left, and more of which we're about to see in a moment, provided we have the mapping on the right, we can identify the exact functional brain networks and brain regions which interact and which are the most or the least, if you cared about that, frequent or most often occurring in our observed data. And then, provided we have the appropriate domain-specific expertise, we can show very actionable and very meaningful conclusions, as well as potentially formulate hypotheses that would drive further studies, and maybe even in the future contribute to translational and clinical science when it comes to, for example, phenotyping, by the title, of neurodevelopmental and neurodegenerative conditions. Here are some of the other most, say, textbook and also pretty informative deliverables, observables of the ELA. Namely, we can compute the frequency visit of the basins, where by basins we mean all the states or all the patterns of activation of the brain regions which share the same attractor, so the local minimum. And please notice that it's not synonymous that some basins are frequently visited with that they contain or are comprised of many states. And to put it intuitively, we could conceptualize it in a way of imagining a landscape that has a basin that's not very deep, so it does not retain the brain or about any dynamical systems in it for too long, but it's very widespread. Another set of metrics, here's the basin graph, which shows us the patterns of transitions from non-attractor states to the eventually attractor states. On the left, we have a exact placement of the nodes, whereas on the right, we have a more approximate and more computationally effective one, but it comes with a nice perk of its own, namely by looking at the layers that form naturally, we can assess what are the average number of steps a brain or system has to take in order to reach the attractor. And these metrics, along with the previous ones, could be very useful when it comes to phenotyping, in the sense that despite them being pretty high level or categorical, if treated as vectors and if quantified beforehand, they can be very potent and actually very informative when it comes to clustering and then binding the clusters of these subjects to specific conditional and symptomatic subtypes. There are certain other extensions of the descriptors, which I have to skip for now, but I'm happy to have a chat about it if anyone's down. Here's a bit about the variational base alternative to the exact formulation of the model, which is a bit more computationally effective and which allows us to quantify the uncertainty of the model. And one other of the major extensions of it is what's called a PDA, so phase diagram analysis, which also comes from statistical physics and tells us things about, for example, state transitions or criticality of any dynamical system, so brain in our case. And please notice that once more, the key metrics and descriptors showcased by the method map very nicely, very neatly to interpretable and biologically meaningful interpretations. let's skip it for now. Here's a proof that we reach meaningful and statistically stable results by ensuring convergence and quantifying the confidence interval. And some of the key promises I mentioned before are the clinical or translational applications of the data, of the methods, whereas the challenges, which I think are just tap perfectly into the big brain themes, are the availability of big data that's actually big, especially for whole brain studies, and appropriate computational measures, because both of these make the methods very demanding. These are the shout-outs I'd like to quickly give for either giving the necessary background when it comes to methodology, or providing me with the data, or just supporting me throughout the work and guiding my development of the project and contributing to it. And as a final, very brief plug of my institution where I had the pleasure of working on the project, we are quite diverse when it comes to the applications and areas we develop. So if anyone's interested to either talk for a bit about the methods I discussed today very briefly and very chaotically, then I'm happy to do so either today or maybe at either of these two events where me and my supervisor, Dr. Cezary Mazurek, will have the pleasure of speaking. And here's a very general link to the website of the center and the emails of the two of us. Thank you.